But no, I was excited to give a talk on this topic because it's a very timely concept. The drugs in this space are really maturing for hepatitis B. Then I realized there was no data to really present on this topic yet. So that made it pretty challenging to put together. So what I thought I would do is really I'm going to focus mainly on the adaptive immune response here and the T-cell response, but try to pull in data and concepts from other models and other diseases that try and maybe raise questions that we should be thinking about in terms of what should be measured as we move forward with these drugs that are really able to reduce HBV antigen in the patients. So let me see, I'm going to make sure that that's working. That's good. So the first thing we want to start with is we all know that there's a HBs antigen burden in these chronic hepatitis B patients. But it's always talked about in terms of units. And being an immunologist, doing these experiments in vitro, we need concentrations, absolute values. And so I really like to put this into perspective. And this is just, the graph here is just showing different categories of hepatitis B patients. And these patients with this kind of E-negative disease, we estimate around 1,000 units of HBs antigen in their serum. And it's pretty straightforward. The serum constitutes about 55% of your whole blood, so it's a simple math problem. So these patients with 1,000 units per mil of S antigen in their serum have about 14,000 micrograms of S antigen in their circulation. So these patients then with the positive disease, you add a zero onto this, and so now you're all of a sudden up to 140,000 micrograms of S antigen in the circulation, 140 milligrams. And so that doesn't even account for the S antigen that's in the liver. So there's a very scientific value that I've given here. That's a lot of S antigen in these patients. And you put this into context of the prophylactic vaccine, which uses 10 micrograms of S antigen to induce an immune response. That is a huge change. It's a huge difference. And it's really important to put these absolute values when we're thinking about the antigen burden. And obviously we all know too that the current treatments with nucleoside analogs, they reduce ALT, they reduce DNA, but they don't change the S antigen levels, even over years of treatment. And we know from mainly animal model studies that this chronic antigen exposure leads to a progressive decline in the T-cell response. You get progressive loss of T-cell function, upregulation of inhibitory receptors, loss of CD4 help, which then leads to a collapse of T-cell response and ultimately deletion of high-affinity antigen-specific T-cells. So virtually all of these aspects have been shown in chronic hepatitis B patients. We know that the virus-specific T-cells, they are prone to apoptosis. They express inhibitory receptors. They have metabolic dysfunction that is consistent with immune exhaustion. And now there's recent data showing that this is also extending into the B-cell compartment too as data comes into the literature. So coming back in terms of looking at the immunological effect of HBV antigen reduction, I think there's really some key questions that need to be addressed as we move forward. So is reversal of immune exhaustion even going to be possible? I think this is likely, but still an open question. But will it just be reducing antigen? Will just reducing antigen exposure be enough to reverse exhaustion? If so, how long will it take? And if you can, what actual immune function? So if you can walk back this kind of inhibitory pathway, how far can you go back? Which immune functions will be recovered if you lose S antigen? And then if immunity recovers, will it actually be effective? Is it going to be too fragile, too transient to have an antiviral response? Or will you have to boost the immune response in conjugation with reducing antigen? So there's some data from animal models, and this is some data presented from an LCV infection model where most of the chronic infection concepts we have, many of them come from. And so in this case, the mice were chronically infected with LCMV, and then they took T cells out and they transferred them either into another setting, which is also a chronic infection, or into a setting that is an acute infection. So either, and then they measured the T cell response. And what happens to the T cell response if they come from a chronic setting and then either stay in a chronic setting or go to an acute? And what they found is it didn't matter whether the cells came from, went into an acute setting or a chronic setting, the T cells from either were able to, the T cells were able to proliferate. But what they found is, interestingly, that the T cells that were coming from these chronically infected animals originally maintained expression of PD-1. And that maintenance of PD-1 expression led to significantly lower cytokine production. So even changing the setting and putting it into an acute model setting, the cells that had transferred still were not as functional as those came from an acute mouse. The other model that I have to show here is a liver-specific induction of T-cell tolerance. In this case, they induce tolerance by expressing the antigen within the liver, and then they transfer these either to another tolerant environment or a non-tolerant environment. And as expected, if you take a naive cell or a memory cell and stimulate them with their antigen, they proliferate quite well. But if you take the T cells that were originally in this tolerant environment, and it didn't matter if you put them back in a tolerant environment or not, and you stimulate them with their antigen, there was no T cell proliferation. So suggesting that that exhaustion was imprinted on those T cells and it wasn't recovered just by putting them into a situation where antigen wasn't present. But what they found, interestingly, in this study was that if you, and this is certainly not something that's clinically possible, but if they lymphodepleted the mice prior to this adoptive transfer, that they induced homeostatic proliferation. And this, the homeostatic proliferation, when you look at the cells, the T cells in this phase, they were able to produce interferon gamma, TNF alpha, so they became functional again. So they, even though they were tolerant when they went in, the proliferation, they filled that niche, and then they became functional. But if you waited until after the homeostatic proliferation came to an end, the cells had re-entered a resting state, you can see that tolerant phenotype was reimposed on the T cells. So that meant that the restoration was transient, only during the time of proliferation. So these are, of course, the mouse models. So what do we know from human data? And I would say data coming from hepatitis C patients right now and SVR after DAAs is probably going to provide some of the strongest data. And so this is some phenotypic data that was provided, given to me by Gehring to present. And this is looking at phenotypic recovery after SVR post-DAA treatment. And you can see that in terms of the PD-1, CD95 is fast, and TIGIT, after SVR post-DAA treatment, you see a downregulation of all these receptors. So PD-1 is also inhibitory and activating receptor. These cells seem to be recovering after HCV is cleared. And then some data from Sonia McPartland and Jordan Feld looking at the functional recovery of T-cell responses after post-DAA treatment or on and post-DAA treatment shows that the breadth of the T-cell response in these Hep C patients continues to increase towards the end of treatment, and then at 24 weeks post-treatment really reaches its maximum, the breadth of the T-cell response, before contracting somewhat at week 80. But of course, this kind of expansion of T cells after SVR is not homogeneous, just like any kind of human study. Some of these patients over the course of treatment show very good recovery of the T cell response, while others do not. And this is going to be a consistent issue that we always have to take into account in these patients. So what about strategies for HBs antigen or HBV antigen reduction in patients? So there was a recently published paper here. Now this is looking at the mouse model. I'll start with this data because this is a mouse, and I'll move into some of the humans. This is antisense oligos, and these behave, I would say, similar to the RNAi that we might be more familiar with, but these are single-stranded DNA molecules that also target the 3' end of the transcripts. They can be targeted to hepatocytes with GalNAc as well, but they work through an RNase H activity instead of the RISC complex. And so in this AAV model, using this AAV HBV mouse model, multiple injections of the antisense oligo led to a significant and dose-dependent decline in HBs antigen, HBe antigen, and HBV DNA. So they had good antigen reduction, but there was no immunology presented in this mouse model. So I think we're also all very familiar now with the RNAi drugs that are coming down the pipeline. In this, I'm using some data from Arrowhead as an example here of a patient that they've presented that received ARC-520 and multiple injections of ARC-520. And you can see that in this study, repeated injections of the RNAi led to progressive decline in HBs antigen, HBe antigen, and HBV DNA. So the patient showed a good response. Of course, this was a single patient that did go on to lose S antigen, and the S antigen decline seemed to coincide to some extent with ALT activation. But again, we don't know if that ALT was an actual antiviral immune response. There was no immunology presented with these patients. And so the other strategy that's also moving forward is the nucleic acid polymers being developed by Replicor. And this is one of their studies using the Rep 2139 and 2165 in E-negative chronic HBV patients. And you can see in the graph on the left, the dotted orange line, the dotted blue line, where they introduced these NAPs into the therapeutic regimen, very significant declines in the HBs antigen levels. And then in the graph in the middle, you see the appearance of anti-HBs as the S antigen declines within the circulation. So, the results from this is that the NAPs are really doing a good job here of reducing the S antigen in most patients. The appearance of anti-S becomes apparent in the serum, but what we don't know thus far is whether that's an unmasking of the anti-S or whether it's T and B cell activation that leads to the de novo production of anti-HBs. So what do we know about S antigen loss and recovery of the T cell response in chronic hepatitis B patients? And Andre showed part of the whole figure of this study previously during his talk. I kind of cherry-picked a little bit of the data to make the example here. And this is a study from Carlo Ferrari's group some time ago looking at nuke-treated patients. And what they showed is that patients that didn't have treatment in the top graph had very low T cell responses after in vitro expansion. But those patients that spontaneously lost S antigen or lost S antigen after nucleoside analog therapy had had significantly increased T cell responses with recovery after nuke treatment demonstrating the highest amount of in vitro T cell expansion. And Andre also pointed out, but this is the issue here is this is a cross-sectional study. And so was it the T cell response that caused the S antigen loss or the S antigen loss that allowed the T cell response to recover? And we don't know that yet. There are a couple of studies in HBV mouse models that have started trying to address this strategy of immune recovery after reducing HBV antigens. And this was the top one here. The first one I'm going to show is a study where they used antibody to neutralize S in the circulation. And then they came in with Engerix B commercial vaccine after many, many injections of the antigen or the antibody. And so in this graph, in the blue line is injection of the neutralizing antibody into these mice. And you can see that when you introduce the antibody, the S antigen goes down in the circulation, the antibody goes away, the S antigen comes back up. The vaccine in the red line does nothing, but then when you combine it, it's difficult to see here, but those, the black line and the black triangles go down and they stay suppressed. So indicating that if you remove the S antigen and vaccinate with the B cell driven vaccine, that you can suppress S antigen in these mice. The problem was they only eliminated S here, and if you actually look, E antigen was still present, and so that means the infection was still ongoing within, well, the infection of HBV was still present in the liver. And they weren't able to actually eliminate that infection until they added a CpG agonist on here to induce a T cell response, really indicating that you need the T cell response to target that reservoir. The other model is a hydrodynamic infection model with HBV, and then they did T cell transfer after this. And to give you a perspective of what this hydrodynamic infection model was, they inject two different plasmids, and in this case, the open squares of the acute infection, whereas the other plasmid induces a persistent infection out to 28 days. And then they took T cells and looked at core antigen-specific T cells here, with the blue being T cells taken from the chronic, the persistent mouse, and the red being from the acute mouse. And what they found was that the T cell expansion was similar between the two, but those T cells coming from the persistently infected mouse did not produce cytokines. And when they adaptively transferred these into a new mouse, or a naive mouse, and gave an acute challenge for HBV, you can see that the blue line the blue line showing T cells coming from a chronically or persistently infected mouse were much less capable of clearing the acute HBV and hydrodynamic transfection in this case, suggesting that that tolerance is maintained. So just to summarize then, the new HBV therapies can effectively reduce HBV antigen concentration in the blood. There's different approaches, clear antigen through different mechanisms. There's either preventing the production, such as the RNAi or the DNA oligos, or preventing secretion, such as the NAPs. And really have to consider this because this may alter the immunological response to infected hepatocytes. So far, I would say there's really no immune recovery data in hepatitis B patients receiving the RNAi or the NAPs. There was evidence of ALT elevation associated with S antigen clearance, but ALT elevation doesn't necessarily equal antiviral immunity, and we really need to address that. The anti-HBs becomes detectable in patients that were given the NAPs, and more work to really focus in on whether that's unmasking existing antibodies or we're actually getting activation of B cells and T cells will be important. And I think the data from these Hep C studies that are coming out will show us that we can at least get partial recovery of the T cell repertoire when you remove that antigen, suggesting that there is a benefit. The animal data indicates that the functional exhaustion is a stable phenotype, or it's imprinted. And so that means that antigen reduction alone may not be sufficient for immune recovery and coming back to this combination with immunotherapies or vaccines. But again, yeah, I think more and more caveats are coming out with these mouse models. We always knew they weren't a mimic of infection, but now the evidence that duration of infection or the amount of antigen is really going to impact these immunotherapy studies in the mouse. We really have to be cognizant of this data. But the fact is, the experiments can and need to be done in chronic patients treated with these new drugs that are getting RNAi and these NAPs. I think this is where we're going to get the best data. So I will finish there and look forward to the roundtable discussion. Thank you.